At the ASH 2025 meeting, we presented some very important updates on Rapcabtagene Autoleucel. We presented a clinical trial evaluating this autologous CD19 CAR T-cell product with a very rapid, short manufacturing time of 2 days in patients with newly diagnosed large B-cell lymphoma that had two important features. Patients either had a high baseline clinical risk based on their IPI being 3 to 5 and or having double hit lymphoma. And after 2 cycles of frontline chemotherapy, they had a partial response or stable disease. So high risk at baseline and then poor response to chemotherapy. And then they could be enrolled in the study single arm trial to evaluate Rapcabtagene Autoleucel. And at this update we presented at the ASH meeting for the first time showing data that Rapcabtagene can result in a very high complete response rate in this double high-risk population in the frontline setting for aggressive lymphomas. Rapcabtagene has some very unique features compared to currently available autologous CD19 products. It's manufactured through something called the T-Charge platform, and this has been previously published for this product in the relapse refractory setting. And the advantages of the T-Charge platform are that it has about a 2-day manufacturing time frame. That's significantly shorter than what we see for other products. And so that has some numerical advantages of potentially being able to get the CAR T-cell turned around faster and back to patients sooner. But it also has some impacts on the actual product that's received. The CAR T cells that are having this rapid manufacturing have increased stemness, have increased proliferative capacity when they're re-infused in the patient, which hopefully will result in an improved efficacy profile and an improved safety profile. What we saw in this clinical trial is that this short manufacturing time frame had a major impact on the timing of toxicities. Instead of having side effects like cytokine release syndrome occurring 1 day or 2 days after a CAR T-cell infusion, in this study, there were rare side effects with cytokine release syndrome that were more than a fever, but when they occurred, they usually occurred about 2 weeks after the CAR T cells were infused, which may allow easier use of this product in an outpatient setting. The safety profile for Rapcabtagene, in my opinion, is very favorable, especially compared with CAR T cells that are available for patients today. The cytokine release syndrome is the most notable toxicity for any cell therapy product. And in this clinical trial, we saw that some patients did have grade 1 cytokine release syndrome, which is a fever, but it was less than 10%, around 10% of patients that had anything more than a fever. So that means 9 out of 10 patients either didn't have cytokine release syndrome at all, or when they did, it was only a fever, which should allow this product to be potentially used much more widely. We also noted that the neurologic toxicities, or ICANS, was seen in less than 10% of patients. So the toxicities of Rapcabtagene in this clinical trial were managed per standard procedures, steroids, tocilizumab, other supportive medications, but thankfully there were less toxicities to manage than what we've seen with previous products in this same space. It's an incredible time to do clinical research for patients with lymphomas because of the wealth of new agents that we have coming along. We had chemotherapy, followed by more chemotherapy, followed by more chemotherapy not that long ago, a decade or so ago. The targeted therapies we had were rituximab, and that was about it. Now we have an avalanche of new exciting agents. And so Rapcabtagene Autoleucel, this rapid manufacturing CAR T-cell, is one of many weapons, but it's one that I'm very excited about, and clinical trials evaluating this in patients with aggressive lymphomas, potentially in patients with newly diagnosed aggressive lymphomas, I think is a very interesting idea, and I look forward to seeing more clinical trials in this space.